Okay, welcome to General Psychology, otherwise known as Psych 101. Today we're going to be talking about biopsychology. So it's chapter 3 of our book. I don't know how far we're going to get, but I aim to cover at least half. We'll see. It's going to be a lot of technical, biological stuff. We're going to talk about all of this stuff. Okay, we'll start with brain imaging techniques. Talk about neurons and the nervous system. Believe it or not, the part about neurons is going to take a while. So the nervous system, we may not get to that until Wednesday, but we'll see. I've written this overview here and it's organized so you can see where it says nervous system, the central and peripheral nervous system. It's organized in a way that makes it easy for you to understand what is contained in what. Okay, because part of this chapter is just knowing the organization of the brain, where things are and what they do, basically. I'm going to try to keep it simple, but it might seem a little bit complicated at times, but just know that the book is even more complicated. All right, so let's start with brain imaging techniques. How do we know anything about the brain? Okay, well, we know things about the brain in various ways, but some of those ways involve brain imaging techniques. Okay, and you can see some images there. The first one here is, this is an image for a PET scan. A PET scan uses radioactivity to produce an image. So basically they have you drink some substance that is actually radioactive but not harmful. And then what happens is the areas of the brain that are more active, that are using more of this energy, and I believe they use radioactive glucose. Glucose is the form of energy your brain uses. So the parts of the brain that are more active are going to appear in red. And then after that, we'd have orange, then yellow, and then, I guess then green, then light blue and dark blue. The least active areas of the brain will appear a darker blue. So that's a PET scan. Now a CAT scan, you see here in the middle, that's a CAT scan. The CAT scan uses X-rays to generate an image, okay? You can see there that it's just like a regular X-ray that they'll use for bones and things like that. It's used to detect tumors and look at brain atrophy as well. You can see the brain there kind of in gray, and if a part of the brain has kind of shrunk, you'd be able to see that. So a CAT scan is used to detect brain structure. So you can look at the structure of the brain. A PET scan is used to look at brain activity. And then an fMRI, which you see here, the image on the right, all the way to the right, upper right, that's an fMRI image. And that uses electromagnetism, and it produces a three-dimensional image, a 3D image, which allows you to basically see both structure and function. So you can see the areas that are in red there or orange. Those are the areas of the brain that are active at the moment. And you can also see the structure of the brain really well. And they do this on a computer, of course, and you can look at the image and zoom into it and go into the brain and look at the inner areas of the brain that you normally can't see, not just the surface. And I want to point out that the machines that are used to produce these images are these huge machines that cost like millions of dollars. It's that machine that looks like a big box, and it has basically a tunnel in the middle, and then they slide you in there and they turn the machine on. Okay. That's what they use for this stuff, for PET scans, CAT scans, and basically fMRI. And they're very expensive machines, but that's how we know something about the brain. We know a little bit of something about how the brain works. That's brain activity, about what it looks like. That's the CAT scan. And the fMRI allows us to see both activity and structure. Let's keep going. Let's just talk about neurons now. What are neurons? So neurons are basically a type of cell that is in the brain. Okay, now you have different kinds of cells in the brain. Okay, you have normal cells like you have all over the body as well. You have cells that support neurons in a way, but we're going to focus on neurons. Okay, neurons, you can call them brain cells or nerve cells. What do they do? Well, they transmit. In other words, they send and receive information. And they also process information. Some of them process information. In other words, allow you to basically understand what it is that is happening. Okay, And they do this in something called the nervous system, which you guys have probably heard of. We'll talk about the, the organization of the nervous system later. And of course, these neurons make many important functions possible. Without these neurons, you're just a bag of meat that doesn't do anything. These neurons allow you to think and feel and remember and walk and talk and do all those things. Without them, you can't really do anything. You're just a lump of flesh, okay? There's a citation there, right? Freeberg, 2010, that's the way you give credit in APA style, remember? So we're gonna start out by talking about these three kinds of neurons. There's three kinds. There are sensory neurons that have to do with your senses, motor neurons, which have to do with movement, and interneurons. And the interneurons are the ones basically that actually allow you to think and to understand things. So sensory neurons, what they do is they gather information in the form of light, sound, odor, taste, and touch. Those are your five senses. The reason you can see something or hear something, smell something, taste something, or touch something, or you understand what you're touching is because of sensory neurons. Actually, you touch something is because of these sensory neurons. So sensory neurons basically, they detect information from the environment. They detect light, sound waves, chemical molecules, pressure, that would be touch, and then taste is also chemical molecules. So they detect these things, and then they send information up to the brain. And that's what I mean by afferent. With afferent, that means the information, the flow of information is basically from the outside, the neurons basically gather information from the outside, and then they send signals to the brain. So the way I remember that is A is for access, so these neurons access the brain. And then we also have motor neurons. Motor neurons control movement in the muscles. Motor neurons allow you to walk and talk and do all the things that involve movement. So here's the thing about motor neurons. They are efferent, okay? And what that means is the information travels from the brain out to the body. And the way I remember efferent is E. It starts with an E. E is for exit. So the information starts at the brain and then exits the brain, goes to the rest of the body to control those muscles so that you can move. And muscles only really do one of two things, really. They contract, which involves energy, right? And then they expand. So you basically, loosen your muscles, and that does not involve energy. That's you basically like letting go, okay? So motor neurons are efferent. The brain really sends signals to the rest of the body so that you can move and walk and talk. For sensory neurons, remember that's afferent. The information travels up to the brain, and then the brain makes sense of it. Interneurons are in the brain. They form circuits in the brain for perceiving, learning, remembering. Interneurons are, you can call them like the really smart neurons. They're the ones in your brain that understand, for instance, what you're looking at or what you're smelling, or the ones that determine, for instance, that you want to walk toward the door so you can leave. The interneurons are the ones that form circuits in the brain, and they allow you to perceive things, understand what you're looking at, to learn, to remember, to make decisions. So you can say the interneurons are really the smart ones. And inter means between. Interneurons actually bridge sensory and motor neurons. So this is the way it happens. Sensory neurons gather information about the environment. That information is sent via sensory neurons up to the brain. The interneurons then basically will analyze or process that information and might basically then understand that we're touching this hot stove right now and that we should move our hand away. The interneurons understand that. And then the interneurons will then communicate with the motor neurons so that you can move your hand away from the hot stove. So it works that way. They work together. They're not really separate. But there's three different kinds: sensory, motor, and interneurons. Let's keep going. Let me check something here. Can't check. Okay, I'm assuming that I'm still recording here. Let's keep going. I hope I'm recording. Here's an image of a neuron that has all the basic parts. They don't all look this simple. But this one has the basic parts. You can see the dendrites there. Okay, they look like branches. And then there's a thing that looks round. That's the soma, which contains the nucleus, and the nucleus contains the genetic information. And then there's this thing that looks like a rod that goes out this way. That's called the axon. And then sometimes the axon has this thing called the myelin sheath, which is just like a covering around the axon. It's like insulation. And then towards the end of the axon, you have these things called terminal buttons. They look like little buttons, like little knobs, and they release chemicals. So dendrites, I'll tell you something a little bit about how it works already and the structure. These dendrites, these things that look like antenna, gather information in the form of chemicals, and then the soma processes that information. And then if the neuron decides that it's going to send a signal to another neuron or to some other cell, it will send a signal which will flow down this way, down the axon. The axon is like a wire. And then there's insulation for the axon. Just like a wire is insulated with a rubber coating, the axon might have insulation, which is made out of something called myelin, which is fatty tissue, around the axon. And that basically helps insulate the signal and helps it travel faster. And it's actually an electrical signal that travels. That's the way neurons communicate, okay? At the end, when the signal reaches these axon terminal buttons, they can then release chemicals that will flow across to the next neuron, okay? And I've told you a little bit about the structure and function of the neuron just by looking at this picture. Let's move on, and let me show you this in words, okay? Just to make it simple for you guys. I'm trying to make it as simple as I can so you understand. So the neuron structure, we looked at the figure already. So remember, the dendrites are those things that look like branches or spikes. They receive information from other neurons. And let me close this. And we have also, let me check I'm recording. Yes, I'm recording. And then we have the soma. The soma is that thing that looks round, that actually looks like an actual cell. That's the cell body. It contains the nucleus and all the other stuff, the so-called machinery for the processes of the cell. So it will contain the ribosome, the Golgi apparatus, all that other stuff that allows the cell to do what it does. And it also contains, you know, like I said, the nucleus with the genetic information. The axon is that thing that looks like a tube that carries information from the cell body to the terminal buttons. And the information is basically electrical. It's an electrical signal that travels down the axon, just like electricity travels on a wire, on a copper wire. Let's keep going. More about the neuron structure. Often the axon is covered by something known as the myelin sheath. Remember, myelin is just fatty tissue, and the sheath is basically just means covering. So the myelin sheath is insulation around the axon. Just like wires have, basically a rubber coating, neurons often have this myelin sheath, this insulation around them. And this insulation does two things. One thing it does, it ensures that the information is not sent to unintended cells. So you can think of that as, you know, making sure that the wires don't get crossed and then gets short-circuited, and then the information, the electrical signal, goes some other place where it was not intended. Okay, that's like when you cross your wires and working with electricity, you cross your wires and you get a basic and electrical shock. Basically means the signal, the electricity went somewhere it shouldn't have gone, and then it caused a problem. Okay, so the myelin sheath, one thing it does, it keeps the signal moving in the right direction. Another thing it does, which is interesting, is it makes the information, the electrical signal, it makes it travel faster. And the way it does that is you have to consider the analogy of a leaky hose. Say you have a hose, and that hose has a bunch of holes in it, and it's connected to the spigot, and then you turn on the water, and what happens is that the water doesn't flow out very fast out the nozzle, because there's a bunch of holes where the water is leaking out, it's spraying out. If you insulate that hose, if you get some electric tape, let's say, and you wrap it around the hose and cover those holes, and then you turn on the water, then the water will flow out much faster out the nozzle. That's kind of what the myelin sheath does. but it's a little bit more complicated than that. But it makes the information travel faster, okay? Terminal buttons are those things that look like knobs or on little twigs at the end of the axon. They will release these things called neurotransmitters. A lot of you have probably heard of neurotransmitters. Neurotransmitters have been described as chemical messengers. Basically, they're chemicals, and they do one of two things. They excite other neurons, so they basically will cause another neuron to fire, or they'll tell the next neuron to fire to send a signal. They will inhibit other neurons. They'll tell the next neuron to stop firing. That's what neurotransmitters do. Okay, they tell the next cell to send a signal or to not send a signal. And it's kind of like a computer, you know, with the code and stuff like that at the most basic level. Zeros and ones. Basically, zero means that, I believe, if I'm remembering correctly, that there's no information flowing, okay? There's no flow of electricity. And the one means, I think, that there is information flowing. So it's like on and off. Excite other neurons, tell the neuron to turn on and start firing, send a signal, or inhibit the other neuron, tell the neuron to stop firing or turn it off, so to speak. Let's keep going. Here are some images of neurons. They're not really, they're more like drawings, okay? You can see over here on the left image, there's a neuron here. You can see some dendrites there, those things that look like spikes, the soma with the nucleus, the axon going this way, and then there's another axon going this way. So this one has like two axons. I did tell you guys that, you know, they can be more complicated. The image that we looked at first, that was a very simple one, but they can have, you know, more than one tail, so to speak, more than one axon. They can have multiple axons communicating, so they can communicate with multiple cells, multiple neurons, more or less at the same time. And then see this axon moves in the direction of this cell over here. You can kind of see a little bit the terminal buttons there, and they can communicate with this cell, and then this cell can communicate with one over here that isn't seen, and then this one over here. This one only has one axon. And they communicate like that. Now, they don't really touch. You'll find in a moment. The terminal buttons don't really touch the dendrites of the next neuron. They don't really touch. But in the brain, you'll have a whole bunch of them, and it might look like something like this. You're born with billions of them, billions of these neurons, and then some of them die. And when you're first developing, it's important that some of them die, and only the ones that are needed survive. But then as you get older, more of them will die. And then when you reach old age, you might have lost too many of them. And then things like remembering and understanding certain things might become more difficult. You might develop symptoms of dementia or even Alzheimer's. Okay, that's caused by several things. But the point is that these neurons form chains and networks and complicated webs in the brain, and they allow for the flow of information. And without that flow of information, you can't remember, you can't think, you can't walk, you can't talk. And of course, symptoms of Alzheimer's, it begins with some forgetfulness. You forget some minor things, okay? Like you forget, you know, when you're looking at the, you know, at the beach, and you're looking at the waves crashing there, and you look out, and you forget what that thing is that's right in front of you. And say, what is that thing called? That's the ocean, okay? You start forgetting simple things like that, and then it gets worse and worse. And then, you know, before you know it, you start forgetting names of even your loved ones. And as it progresses, it gets worse and worse. Eventually, you forget how to talk and how to walk. And before you die, when you have Alzheimer's, you basically have forgotten like almost anything. You're just sitting there like a lump. You can't walk, you can't talk, you can't feed yourself, you can't do anything anymore. And it progresses, gets worse and worse. Luckily, there are treatments that are being developed for Alzheimer's, and so people can live longer with the disease now, and you know, basically reduce the severity of symptoms, but there's still no cure. Let's talk about, so that was the neuron structure and what they look like. Now let's talk about how the neuron functions. Let's talk about how it works. And I'm not gonna get into too many technicalities. I'm gonna make it as simple as I can for you guys. So first we'll start with the neuron at rest. So the neuron at rest, it hasn't yet sent a signal. It hasn't done that. okay? So it is said to be at rest. It's not firing, it's not sending a signal. And the important thing to note is that at rest, the neuron has what is called a resting potential, an initial charge of -70 millivolts. So the neuron that is not sending a signal, that is not firing, has an electrical charge, and it's a negative charge. -70 1,000ths of a volt. That's the charge of a neuron when it's not sending a signal. And when the neuron is not adequately stimulated, let's say something happens, but you didn't notice, you didn't feel anything. Let's say an eyelash or a little piece of lint or a hair falls on your skin, and you didn't even notice, you felt nothing, which means your brain didn't get a signal, okay? So there's some kind of light touch, eyelash falls on your skin. What happens is that you get a graded potential. There are neurons on your skin that have that resting potential of -70 millivolts, and when they detect that eyelash falling on the skin, there might be a small change in the electrical charge. So it might go from -70 millivolts maybe to -68. So it changes a little bit toward the, toward the positive end. There's a small change in the electrical charge, and then it just fades away. It goes back to -70. The neuron doesn't fire, no signal is sent to your brain. You felt nothing, you know nothing about that eyelash, being on your skin. When the neuron is adequately stimulated, let's say there is something that you do feel, you noticed something, you heard something, you felt something, that's when the neuron is adequately stimulated. So you get a hard slap, for instance. It doesn't have to be a hard slap. It could just be somebody grabbing your arm or pinching you, or just a light touch. That's enough for you to notice, okay, for your brain to get the signal. What happens is that the neurons on your skin will go from having a charge of -70 millivolts, and then the voltage will rise by 5 or 10 millivolts. It'll go from -70 to -65, maybe -60, and then once it changes by at least 5 or 10 millivolts, that will get other things in motion. So that's called the threshold of excitation. That is a magic number there. You change the voltage by at least that much, and then the neuron will it'll set things in motion, and the neuron will eventually fire. So that rise in the voltage at 5 or 10 millivolts causes a chain reaction. By the way, there are things here in bold that you have to know, like -70, that's another, -70 millivolts is another magic number that you have to know there. That's when the neuron is at rest. You have to know what a graded potential is, so I'm trying to keep things in bold that you should know about. So once you get a rise in the voltage by at least 5 or 10 millivolts, you reach the threshold of excitation. In other words, that's when things get started, and that leads to a chain reaction that results in the neuron firing. Let's keep going here. The neuron in action. That chain reaction results in what we call an action potential. An action potential, okay? Something that you need to know. An action potential is when the voltage rises from -70 all the way to +30 millivolts. So when you get a change of at least 5 or 10 millivolts, that's the threshold of excitation, the voltage will keep rising, and it will pass 0, and it will go then to 10 to 20. When it gets to +30 millivolts, so now you have a positive electrical charge, that is called depolarization. That is when the neuron fires. That is when it sends a signal. The voltage after that will fall back down to -70 millivolts. It will return to its resting potential. So the action potential is basically the voltage rising all the way to +30, where the neuron fires, and then it falls back down to -70 so that it can fire again. That is the action potential. It's basically the rise of the voltage to from -70 to +30, and then back down to -70. That whole sequence is called an action potential. And remember, at +30, that is another magic number. That is where when you get to that voltage, the neuron fires. It sends a signal. All or none response means that the neuron fires at full strength, or it doesn't fire at all. The neuron has been said to be like a loaded gun. Okay, if you squeeze the trigger just a little tiny bit, and then you let go, the gun won't fire. That is like a graded potential. But if you squeeze the trigger just a little tiny bit more, what will happen is the gun will fire, and it will fire at full strength. You just need to squeeze it just a little bit and it will fire. And if if you squeeze it again, it will fire again, and it will fire the same way every time. The neuron always does things the same way. It's the all or none response. Voltage starts at -70, rises all the way to +30 if there's enough stimulation, falls back down to -70. It actually, believe it or not, it overshoots a little bit and goes back down too far. It goes to like -75, and then it has to correct itself to get back to -70. And that's a little bit more information than you need to know for this class. But by the way, what's causing the change in the voltage are things, ions, that have positive and negative charges that flow in and out of the cell, in and out of the neuron, basically. And the concentration of those ions, those positive charges and negative charges, is what determines this voltage. Okay, so the neuron always does the same thing every time. Okay, all or none response. It's also been described as being like a toilet, right? You pull that little lever. If you pull it just a little bit, you'll hear a little bit of water flow, but then you let go and nothing will happen. It won't flush. That's like a graded potential. Pull the lever just a little bit more and the toilet will flush properly, and it will flush completely and then refill itself so it can flush again. That's the way the neuron works. And by the way, the toilet analogy, that's appropriate if your toilet is working properly, okay? It's also been said that the neuron works like, it's also like an orgasm. You get some stimulation, right? But not enough. That's like a graded potential, no orgasm. But if you get just a little bit more, then you can't control it, and then you orgasm, okay? And then you go back to normal, and so that you can have another orgasm later. But in this case, it might take some time. Okay, those are all analogies that have been used to describe how a neuron works. Let's keep going. Here's what it looks like visually. So we start with a resting potential here at -70 millivolts. That's the membrane potential. That's basically the charge across the kind of the membrane or the skin, you could say, of the neuron. Their concentration of positive and negative charges in and outside the cell determines the voltage. So it starts at -70, and then there's a slight change in the voltage, change of at least 5 or 10. You reach the threshold of excitation, basically, and then the voltage keeps going all the way up beyond 0 voltage, in which there is no voltage, all the way to +30. And then the voltage falls back down. It goes too far at first. You get hyperpolarization. The voltage goes to, it goes to like -75, -78, somewhere around there, millivolts, and then it has to correct itself. And the flow, right here, it's the flow of those positive and negative charges, those ions, which you get, by the way, from the things you eat. That's what determines the voltage, the flow in and out of the cell of these ions, these positive and negative charges, basically. Like when you eat, like salt is sodium chloride. Sodium is positive, chloride is negative. The relative concentration of those things, and calcium is positive, and other things, potassium positive, those things, the relative concentration of those things in and outside of the cell determine the voltage. And things flow into the cell and things flow out, and that causes changes in the voltage, and it causes this change that we see here. Okay, and that should remind you guys of something. You know, this little spike, if you see it over and over again, it kind of looks like an EKG, right? You see the little spike, they're measuring your, the activity of your heart. That means you're still alive, your heart is still working. There are neurons that communicate with your heart and tell your heart to keep beating, tell your heart basically to keep working. If the heart stops receiving signals, it will stop and then you die. There's a little bit of neuron simulation here. You guys can click on that and play with that. I don't know if it's gonna work. Probably won't work on my computer because it's brand new. I probably don't have what I need in order to play it. So I'm not gonna mess with it, but you guys can play with it on your own. You'll see the voltage go up and down right there and form that little spike. More about neurons and how they communicate. I told you guys that neurons communicate with these things called neurotransmitters. Neurotransmitters are chemicals that are released by neurons that regulate important functions. Okay, here are some of them. Serotonin, which regulates mood, sleep, and hunger. If serotonin levels are too low, you feel awful, okay? You feel really sad and depressed and awful. So that's how it's related to mood. If serotonin levels are high, you feel much better. Serotonin levels are also related to sleep and hunger, as is depression. When you are depressed, serotonin levels are low, and you also may have trouble sleeping, and hunger also seems to be less, okay? Often people don't feel very hungry when they're depressed. Their serotonin levels are low, and they don't even seem to enjoy food. So serotonin is related to those three things: mood, sleep, and hunger. And the way to remember that is, well, just remember depression, and depression affects all those things, okay? Dopamine is another commonly known neurotransmitter. It affects mood, sleep, and learning. So dopamine also affects mood. When you have higher levels of it, you also feel good. You feel a sense of reward, right? Also affects sleep and learning. If there's not enough dopamine, it's hard to form memories, and well, you can't remember things very well. Acetylcholine is another neurotransmitter. It's involved with muscle action and memory. Acetylcholine, yeah, is needed for muscle action. If you run out of acetylcholine, you become paralyzed, and then you stop breathing and you die, because basically your heart won't pump, your lungs won't expand. You'll die. Okay. If I remember correctly, I think the venom of a black widow spider causes too much acetylcholine to be released. Let's say a black widow spider bites a little bird, right? Too much acetylcholine will be released, and then the bird will start twitching and spasming, right? And then after a while, it runs out of acetylcholine, and the bird becomes paralyzed and dies. And then the black widow spider can eat that little tiny bird. I guess it would have to be a tiny bird. If you get bitten by a black widow spider, that probably won't happen to you. I mean, they are dangerous, but not enough venom to really, for you to die. But it is dangerous, the venom of a black widow spider, for those who are young and those who are old. Okay, so keep that in mind. Norepinephrine is another, acetylcholine is also related to memory, okay? Without acetylcholine also, it's harder to form memories. Norepinephrine is involved with arousal and alertness. There's an easy way to remember norepinephrine. Norepinephrine is the same thing as adrenaline, or epinephrine. Epinephrine is the same thing as adrenaline. Norepinephrine is the same thing as what we call noradrenaline, okay? When it's used in the brain, it's called norepinephrine, okay? Or noradrenaline. When it's in the body as a hormone, okay, then it's called epinephrine or adrenaline. And what that does is that it increases alertness, arousal, increases breathing. You can run faster, hit harder, right? That's what norepinephrine is for. But it's not just during times of an emergency, it's also during normal waking hours, right? You have norepinephrine running through your body and your brain to keep you alert and awake and for you to do the things you do. If that will increases dramatically, that's during an emergency, and then you could kick things into high gear, so to speak. Glutamate is another neurotransmitter. Glutamate is the main excitatory neurotransmitter that allows the neurons to actually send signals to each other, to fire. Without glutamate, the neurons cannot send signals to each other. So it's involved in the excitation of those neurons. It's also involved with learning, long-term memory as well. I don't know why the screen got a little darker. GABA is involved with brain function, sleep inhibition, and mood. Let me explain GABA. GABA is released when you consume alcohol. Well, GABA is also created naturally in the brain, but alcohol increases GABA in your brain. And we know what alcohol does. Alcohol slows you down. Neurons are not firing as much, or they fire more slowly. If you have too much alcohol, and then before you know it, you can't move very well, you can't remember very well, you can't think very well, and then you end up doing stupid things when you're drunk. That's because of GABA. GABA reduces brain function by slowing down the rate of firing among those neurons, reducing the firing in general, and then it starts to affect the brain. Too much GABA, of course, and you pass out. And that's what happens when you have too much alcohol. And if you get GABA even beyond that, if you get way too much GABA, you will die. You will stop breathing and die. And that's what happens when you drink too much alcohol. You can pass out, and that's like, you can think of that as your body protecting you, so you can't drink anymore. But if you drink alcohol very, very fast, you can drink beyond the point of blacking out. And you may black out, but you may also stop breathing and die. Beta endorphin, you guys know that as just endorphins. They're involved with pleasure and pain. More endorphins means you feel better, you feel pain. I mean, you feel better, more pleasure, less pain. There are many other neurotransmitters. These are just some of the common ones. There are dozens of them and dozens of them. And they're discovered more and more of them every now and then. A little bit more about about dopamine. Dopamine plays a role in addiction. Some of you may be wondering about addiction, right? Addiction is partly related to dopamine, okay? Because when you get dopamine in your brain, it makes you feel really good. And then you want to feel good again. And then you want more dopamine is what happens. Grossman in 2015 published this report that says that, these app games or these games that you guys play, video games, they take advantage of the actual app games. Let's just stick to the app games, the ones on your phone, on your tablets, and even on your TV. They take advantage of something called a compulsion loop. So they get you hooked and they get you addicted. And the way it works is this: you play the game and you conquer a certain level. You feel good because you passed that level. And then what happens is you also get rewarded with coins. These fake virtual coins or little dollars or something like that, usually coins. And then you feel good. You earn some coins. You can use those coins to buy things to help you pass the next level. Maybe you buy more armor or better weapons or whatever it is. But you get rewarded with these coins, you feel good, and then you buy things to help you pass the next level. And then you pass the next level and you get rewarded again. And you get that dopamine, that rush of dopamine again. Every time you go around the loop, your brain gets this little burst of dopamine, which gives you a sense of reward, a sense of satisfaction. So you play a game, you conquer a certain level, you feel good, earn some coins, get rewarded, you get some dopamine. Then dopamine levels go back down and you play the next level, you pass the next level, right? And you feel good, you get reward again. And you keep getting this reward. You keep going on this loop. Playing, getting coins, getting rewarded, playing again, getting coins, getting rewarded again. And it gets you addicted, it gets you hooked if you do it long enough. Each time you feel good, your brain is getting a little burst of dopamine. And it's the same thing that happens when you go shopping and you shop for something and you feel good afterward. Or you get a new car, or maybe a new house, or you meet someone, you get this burst of dopamine and you feel good. But then afterward, dopamine levels will go down again and you'll feel just as miserable or the same way you did before. And then you want to do things to keep, that dopamine coming, to keep feeling good. And that's why we do the things we do. We keep shopping, we keep playing games, we keep doing the same things, trying to continue to feel good, trying to get that burst of dopamine. More about dopamine. There's something known as the dopamine hypothesis. It doesn't say there, but that's what this is, dopamine hypothesis. And I'm just going to summarize this for you. But what this says is that basically high levels of dopamine basically lead to symptoms of schizophrenia. High levels of dopamine result in something called psychosis, which means you become out of touch with reality. Okay, that's what happens with high levels of dopamine. It's exactly what you get it's exactly what you get when you're schizophrenic. You have similar things happening. You're psychotic. You are out of touch with reality. And when you take drugs like L-dopa, methamphetamines, cocaine, they will also increase your basically levels of dopamine. If levels of dopamine are too high, you not only feel good, but you can, if they go higher than that, then you don't feel good. You become psychotic. You start, acting crazy basically. Normal levels of what you need for normal activity and normal functioning. If dopamine levels are too low, please have your microphones off. If dopamine levels are too low, then you get problems with movement. Okay, motor disturbances. You get problems with movement where movement doesn't work very well. You get symptoms of Parkinson's disease, okay? And drugs like, that are called dopamine antagonists, which reduce the level of dopamine, like phenothiazines, will cause problems with movement. So that's called the dopamine hypothesis. You want normal levels of dopamine, too high, and you become psychotic, like a schizophrenic, too low, and you become like someone who has Parkinson's disease, and you have problems with movement. More about neuron communication. The neurotransmitters, remember I said that they have to flow across to the next cell to affect the next cell, right? To tell the other cell to fire or not fire. Well, it turns out the neurotransmitters have to cross a little gap. That gap is called a synapse. The terminal buttons are not touching the dendrites of the next cell. The neurons aren't really touching. There's a gap, okay? And that gap is what the neurotransmitters have to cross, okay? So the small gap between neurons is called a synapse or a synaptic gap. It's also called the synaptic cleft. The neurotransmitters from the terminal buttons are held in these things called synaptic vesicles, and then they fuse with the outer layer over here of the terminal buttons, and then they are released, and they flow across to the next cell, where they can excite the next cell or inhibit the next cell. And what these neurotransmitters do is they will attach themselves to these, basically, what are called these receptors. And when the right one attaches itself to the right receptor, it can open up gates or channels to basically let those, let other things into the cell. Other things that will affect the voltage, that will make it more positive or negative. And then that's how the neuron then works. The neuron can then fire, or let's say it cannot fire. It can stop firing, or it might even be less likely to fire if the voltage changes in a certain direction. So it's been said that neurotransmitters work like a lock and key. If the right one attaches to the right receptor, to the right place, then, gates will open, things will open that will let things into the cell. Things can come into the cell and things can flow out of the cell, and that will affect the voltage. Okay, that's kind of how they work. Okay, the next part is about the nervous system. So I will actually stop there and leave that. That's the second half. I'll leave that for next time. And I will stop recording. Hold on, you guys. Trying to figure out how to end this. Okay, let's stop recording.